My name is Anna Sun, and today I will be discussing potato de-etylation. For this experiment, I left the potato in the dark for about 2.5 months. After, the potato was taken out and left in sunlight. One picture of the potato was taken each day for 8 days. This video will include images of the potato over the course of 8 days, along with a discussion of the observations. Afterwards, an explanation of the results will be given. On day 1, when the potato was first taken out of the dark, the potato had grown pale white stems. The process of growing these stems, or tubers, is known as etiolation. Etiolation is a useful adaptation because potatoes that usually grow in the dark would usually use these stems to break through soil and reach sunlight. This way, photosynthesis can occur and food for the plant can be made. Because potatoes grow underground, there is no sunlight and photosynthesis cannot occur. So, in order to conserve resources, leaves and chlorophyll, which are usually used in photosynthesis, are absent from the etiolating potato, as seen in the picture. On day 2, the tubers of the potato had become a more opaque white and also exhibited a more yellowish tinge than the previous day. The potato tubers on the 3rd day had become a greenish yellow. The green was most concentrated where the tuber was closest to the light source. This is because the parts of the tuber closer to the light had more light to use for photosynthesis, resulting in the production of more chlorophyll and hence more of the green color that we see on the tall stem to the far right. As you can probably tell, the middle stem of the potato is now gone. Before I took the picture of day 4, I had bumped the table. The potato fell off the table and the middle stem came off. Anyway, as we can see through the picture of day 4, the green pigment has now spread all over the potato tubers. It is no longer only present at the topmost part. However, most of the green pigment is still concentrated there. In addition, the stem starts to lean towards the light source, which is on the top left. When we put day 3 and day 4 pictures side by side, the huge change in the angle of the stem overnight can be seen. This change occurs because the plant is trying to reach light, again so it can photosynthesize and make sugar for energy. Being able to detect and grow towards light would be a useful adaptation for potatoes because once they break through the soil, they can grow closer to the sunlight and synthesize as much glucose as possible. On day 5, the whitish yellow color from the tubers are almost completely gone, and now the tubers are a solid green color. On day 6, we can see the potato tubers turn a more darker green, and besides that, there aren't many other changes. Again, we can see on day 7 that the potato tubers have grown a little darker in color, however, besides that, there are no other changes. The potato tubers grew a little darker in shade on day 8, but besides that, there were no major changes. Now let's look at the tiny stem to the very far right of the potato. We can see that it is a much lighter green than the stems to its left. So why is this? Well, we know that the light source comes from the top left corner. We also know that photosynthesis can only occur when sunlight is present, because light is used as energy to essentially help fuel a process. More specifically, light is used to excite electrons in the photosystems so that the electrons can be pulled off and be used to perform chemiosmosis, in which the electrons move down their concentration gradient and create energy, which is used to transport protons, so that they can create ATP, a form of energy. So, back to the picture. If the stem is farthest away from the light, it has the least amount of light available for photosynthesis to occur. In addition, we can see that the taller tubers next to the small one block sunlight and shade the small stem. Since there is less light and not as much photosynthesis can occur, less chlorophyll is present. Chlorophyll's job, after all, is to absorb energy from light. When there is little light to absorb, only small amounts of chlorophyll are required to do the job. Chlorophyll, again, is what gives plants that green pigment, and so it is reasonable that the little stem farthest away from the light would be the least green. An observation that I failed to make until the 8th day was the small budding leaves that were beginning to grow from the tubers. Although the pictures are a bit blurry because I had to take them very close up, we can still make out the beginnings of leaves that are sprouting at the circled marks. This observation, along with others noted, will be explained shortly. So, how did the potato in question change from having pale white tubers to having dark green tubers with leaves? In short, the potato detects an external stimulus, the sunlight, and responds to it. This response includes observable effects such as greening stems that stop growing, as well as the production of green leaves. These changes are part of a process known as de-etylation, the focus of this video. De-etylation can be divided into 3 steps: reception, transduction, and response. In reception, signals are detected by specialized proteins that change shape when presented with a specific stimulus. These proteins are called phytochromes. These receptors are very sensitive and respond to even the smallest amount of light, which is why it was crucial for the potatoes to be left in complete darkness while they were etylating, so they could etylate properly. In the second step, transduction, small molecules called second messengers deliver the message from the light-activated photochrome. These second messengers amplify the signal and leads to the production of specific proteins that correspond with that signal. In the third step, response, a signal transduction pathway leads to responses of the cell in which cellular activities are regulated with the use of enzymes. In de-etylation, some enzymes that are produced are used in making chlorophyll and helping with photosynthesis. This explains the production of the green pigment. Leaves are used in photosynthesis, and so these types of enzymes are also responsible for the initiation of leaf production. This explains the leaves that we saw in the pictures. Other enzymes created through de-etylation regulate plant hormones. Auxin, the hormone that enhances stem elongation in the dark, are less prevalent when de-etylation occurs because the enzymes produced in the process lower auxin concentrations. This explains why the tubers stop growing after being exposed to light. In conclusion, etylation occurs when a potato is left in darkness. Pale, bare, white shoots grow in order for the potato to maximize its chances at reaching sunlight. When the potato is exposed to light, the light activates a photochrome, which in turn activates a signal transduction pathway, ultimately leading to the production of enzymes which help to regulate plant functions. These enzymes lead to physically observable traits, such as greening of the stems and the appearance of leaves. And that's the end of my video. Thanks for listening!